everyone. I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before starting the topic, please like, share, and subscribe the channel. In the last session, we studied about properties, ampholyte and isoelectric point, Sorensen's titration, general reactions of amino acids. In today's session, we will continue with the same. Ester formation by the OH group. The hydroxyl amino acids can form esters with phosphoric acid. In this manner, serine and threonine residues of proteins are involved in the formation of phosphoproteins. Similarly, these hydroxyl groups can form O-glycosidic bonds with carbohydrate residues to form glycoproteins. In this diagram, we can see that phosphoserine is formed from serine in ester formation by OH group. Reaction of the amide group. The amide groups of glutamine and asparagine can form glycosidic bonds with carbohydrate residues to form glycoproteins. In this diagram, we can see when glutamine is formed from glutamic acid in reaction of the amide group. Reaction of the SH group. Cysteine has a sulfidyl group and it can form a disulfide bond with another cysteine residue. Two cysteine residues can connect to bond polypeptide chains by the formation of interchain disulfide bonds or links. The dimer of cysteine residues is sometimes called as cysteine or dicysteine. In the diagram, we can see cysteine is formed by two cysteine molecules in the reaction of SH group. Amino acid derivatives of importance. Gamma aminobutyric acid, that is gamma, which is a derivative of glutamic acid, and dopamine, which is derived from tyrosine. Both are neurotransmitters. Gabapentin, as an analog of GABA, can pass through blood-brain barrier and can form GABA in the brain. Gabapentin is clinically used to relieve pain. In this diagram, we can see that gabapentin can pass through blood-brain barrier and can form GABA in the brain. Um, Histamine synthesized from histidine is the mediator of allergic reactions. Tyrosine, Thyroxine derived from tyrosine is an important thyroid hormone. Cycloserine, which is derived from serine, is an antituberculous drug. Azathioprine inhibits where reactions where amide groups are added and so acts as an anti-cancer drug. Histidine residues are important in the buffering activity of proteins. Ornithine and citrulline are derivatives of arginine and are essential for urea synthesis. In this diagram, we can see that ornithine and carbamoyl phosphate are converted to citrulline in presence of enzyme ornithine transcarbamylase. Peptide bond formation. Alpha carboxyl group of one amino acid with reacts with alpha amino group of another amino acid to form a peptide or CONH bridge. Proteins are made from polymerization of amino acid through peptide bonds. In this diagram, we can see that peptide bond is formed by joining two amino acids. Color reactions of amino acids and proteins. Ninhydrin reaction. Amino acid plus two molecules of ninhydrin give aldehyde with one carbon atom less plus CO2, that is carbon dioxide, plus purple color complex. The amino acids, when treated with ninhydrin, can form complexes pink, purple, or blue in color. In this diagram, we can see that amino acid combines with two molecules of ninhydrin to form a purple color complex. In case of proline, it gives yellow color complex. In the diagram, we can see the test tubes containing negative and positive ninhydrin test. In case of proline, yellow colored complex is formed. The color complex is called Rohman purple. Proline and hydroxyproline will give yellow color with ninhydrin. Amino acids with amide group, that is glutamine and asparagine, produce brown color. The ninhydrin may be adopted for both quantitative as well as quantitative estimation of amino acids. It is often used for detection of amino acids in chromatography. Proteins do not give a true color reaction, but the terminal end amino group of protein will also react with ninhydrin to produce a blue color. Biuret reaction. Cupric ions in alkaline medium form a violet color complex with nitrogen of peptide bond. This needs a minimum of two peptide bonds, and also individual amino acids and dipeptides will not answer this. This In this diagram we can see the reaction of Biuret test. Peptide bonds combine with cupric ions to form peptide copper complex. In this diagram, we can see test tubes showing biuret test. Positive test shows deep purple color. This reaction can be used for quantitative estimation also. The name is derived from the compound biuret, which is a condensation product of two urea molecules, which also gives a positive color test. Magnesium and ammonium sulfates interfere with this reaction. Xanthoprotic test. The ring systems in phenylalanine and tyrosine and tryptophan undergo nitration on treatment with concentrated nitric acid when heated. The end product is yellow in color, which is intensified in strong alkaline medium. This reaction causes the yellow stain in skin by nitric acid. In this diagram, we can see that tyrosine or tryptophan can, when heated with concentrated H10H3, forms nitrated product yellow color complex. In the diagram, we can see that a positive xanthoprotic test shows a yellow color complex. Meron's test. The phenol group of phenylalanine and tyrosine containing proteins, when heated with mercuric sulfate in sulfuric acid and sodium nitrite, or mercurous and mercuric nitrates in nitric acid, form red color mercury phenolate. In the diagram, we can see that a tyrosine, when heated with mercuric sulfate in sulfuric acid and sodium nitrite, form brick red color complex. In this diagram, we can see that a positive Millen's test shows a brick red color complex. chloride interferes with this reaction, and so it is not suitable to test for tyrosine in urine samples. Both xanthoprotic and Mirren's tests are negative for tapioca, that is cassava, which is deficient in phenylalanine and tyrosine. Aldehyde test for tryptophans. In the Hopkins score test, tryptophan contains protein mixed with a glyoxylic acid, and the mixture is laid over a concentrated sulfuric acid. A violet color ring at the surface of liquids shows the presence of indole ring. In this diagram, we can see the when tryptophan combines with glyoxylic acid, and the mixture layered over concentrated sulfuric acid forms purple colored indole ring. In this diagram, we can see the purple colored ring is formed. Formaldehyde and mercuric sulfate is used in similarly in Acre-Rosenheim reaction to get a violet color. Para-dimethyl amino benzaldehyde and strong hydrochloric acid will give dark blue color, that is Ehrlich reaction. Gelatin with limited tryptophan content will not answer the test. Sakaguchi test for arginine. Free arginine or arginyl residues in proteins with alpha naphthol and alkaline hypobromide is to give bright red color. This is due to the guanidinium group. In this diagram, we can see free arginine or arginyl residues in proteins react with alpha naphthol and alkaline hypobromide is to give bright red color. In this diagram, we can see that a positive Sakaguchi test shows a red color complex. Sulfur test for cysteine. When cysteine or cysteine containing proteins are boiled with strong alkali, organic sulfur splits and forms sodium sulfide, which on addition of lead acetate produces lead sulfide and as a black precipitate. In this diagram, we can see that amino acids containing sulfur show black precipitate. Methionine does not answer this test because sulfur in methionine is in thioether linkage, which is difficult to break. Albumin and keratin will answer the sulfur test positively, but casein will not give a negative test. Nitoproside reaction for SH groups. Proteins with free sulfhydryl groups, which gives a reddish color with sodium nitroproside in ammoniacal solution. Many proteins give a negative reaction in the native state, but when denatured, a reaction will be positive, showing the emergence of SH group. Here in this diagram, we can see that no reaction in the native state, but when denatured, reaction will be positive, showing the emergence of free SH groups. In this diagram, we can see cysteine gives red color complex. Pauli's test for histidine and tyrosine. Diazobenzene sulfonic acid with imidazole group of histidine to form a cherry red color complex. diazotized product under alkaline conditions. The same reagent will give a orange red colored product with phenol group of tyrosine. In this diagram, we can see diazobenzene sulfonic acid reacts with imidazole group of histidine to form a cherry red colored diazotized product under alkaline conditions. The same reagent will give an orange red colored product when phenol group of tyrosine. In this diagram, we can see that histidine and tyrosine give a red colored complex. Here ends our session. Kindly like, share and subscribe the reaction channel. Thank you.